Hello, everyone, and welcome to today's webinar titled Characterization of Anti-HER2 Antibodies Using Advanced Flow Cytometry and BLI Label-Free Detection. This webinar is part of the 6th annual event in the Drug Discovery and Development Virtual Event Series. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, go to sartorius.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during this presentation. You can find that live chat located on the left of your screen. You can also participate by submitting as many questions as you'd like during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of this presentation. Now, if you have any trouble seeing or hearing this presentation, just simply click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract tab from the menu at the left of your screen and follow the process to obtain those credits. Without further ado, I now present today's speaker, Kirsty McBain, a scientist at BioAnalytics Applications Group at Sartorius. For a complete biography of our speakers, please visit the presenter tab from the menu at the left of your screen. Kirsty, welcome. You may now begin your presentation.
Today I'll be presenting this webinar focused on the characterization of antibodies, entitled Characterization of Anti-HER2 Antibodies Using Advanced Flow Cytometry and BLI Label-Free Detection. So first on the agenda is an introduction to the instruments that were used to generate the data in this webinar, which were the iQue Advanced Flow Cytometry platform and the Octet BLI Label-Free Detection system. I'll then outline the antibodies that were used to exemplify this combined workflow approach to antibody characterization, and then I will go into a description of the assays that were used, along with some specific data examples. So this is the iQue Advanced Flow Cytometry platform. One of its key features is its very high speed at acquiring sample, and this is due to the patented air gap sampling technology, which means that the probe can move very quickly between the wells of a 96 or 384 well plate without the need to flush the sample in between the wells. So this obviously drastically speeds up the rate of acquisition. The iQue has 15 detection channels, so that means a vast quantity of information can be collected in a single run. The volume requirements for the iQue system are very low, and also particularly with many of the reagent kits. So typically the required volumes for those are about 5 to 10 microliters. It's also very easy to use, largely due to the inbuilt Foresight software, into which you can import things such as analysis templates from the preset templates in the kits. And then these will auto-populate during sampling. They will also be accompanied with preset compensation values, so this drastically speeds up the time to for assay development and also data analysis. And then this just shows some examples of the kits in the iQue portfolio. So these cover a range of different disease areas, namely in immuno-oncology, antibody discovery, and foundational cell functions. And this is the Octet BLI label-free detection system. So it's largely based around these dip and read biosensors, which include a 2D binding surface onto which there are specific molecules immobilized depending on the type of sensor. And then this will allow interactions with soluble molecules in your assay plate to be measured. And this can be used to measure a range of different parameters. So for example, you can look at the binding specificity, so just an indication of whether molecules interact with each other. It can give information on binding kinetics. So the software can generate values such as association and dissociation rates. Those association and dissociation rates can then be used to generate a measure of binding affinity. So for example, KD values. And then also the Octet can be used, for example, to look at concentration analysis. So for example, to determine the titer of IgG in a sample. And this is done by measuring the binding rate of your sample and comparing it to the binding rate of a known standard curve. And this is just a list of some of the off-the-shelf biosensors that are available for the Octet. So each has a different molecule immobilized on its 2D binding surface and means that it can be used for different applications. So for example, if you wanted to quantitate IgG, you could use either the protein A, protein G, or protein L biosensors. Or if you wanted to measure the kinetics of antibody binding, as I'm going to show some examples of later in this webinar, you could use the anti-IgG Fc capture biosensors or the AHC biosensors. Or another one I'm going to show some examples of, which gives you a bit more flexibility in terms of what proteins you immobilize onto the sensor. You could use some in the range of the streptavidin-based biosensors. So obviously this means you could immobilize any biotin-tagged protein that you have in order to monitor the interactions with other molecules. And then another example I'm going to show later, which isn't included in this list, are from the GlyM and GlyS kits. So these contain everything you need from protocol to buffers to the biosensors in order to measure the glycan content of either of your protein of interest. So this would be specifically mannose or sialic acid. And these are the antibodies that were used to exemplify this workflow. So we've used 3 anti-HER2 antibodies. The first is a trastuzumab biosimilar. So this is an anti-HER2 human IgG1 antibody with a variable region targeting HER2 via domain 4. Then we've also had Kadcyla. So this is an antibody drug conjugate, which is based on trastuzumab, with the real key difference compared to trastuzumab being that it has mutations in its variable region to link to that ADC payload, which in this case is tanzine or DM1. And obviously, because this antibody is also based on trastuzumab, it's going to target HER2 via that same region on domain 4. And then finally, we have the pertuzumab biosimilar. So this is another anti-HER2 IgG1, except with a different variable region, which targets a different epitope of HER2 in domain 2. And so we split the project up into a multi-stage workflow, determining different characteristics about the antibodies at each phase. So in phase 1, we really focus on the binding of the antibody to the antigen. And we did that on one hand by looking at binding to live cells using the iQue platform. So doing this, we could do things like generate concentration response curves in order to compare relative EC50s of the antibodies. We could also include both antigen positive and antigen negative target cells in order to get an idea of the antibody specificity. We then, in parallel, used the Octet to have a look at the kinetics of antigen binding. So this allowed us to measure the association and dissociation between the antibodies and a soluble HER2 protein. And then the software could use those dissociation and association rates to generate KD values. Next, in phase 2 on the iQue, we had a look at the competition of binding, so we could get an idea of which antibodies bound to the same or different epitopes. And then for phase 2 on the Octet, we had started to have a look at post-translational modification in the form of that glycosylation using those GlyM and GlyS kits that I introduced earlier. So we can measure the mannose and sialic acid content to get an idea of the stability and also the Fc function of the antibodies. Then on phase 3 on the iQue, we took a real deeper dive into the function of the antibodies by looking at using the cell-based assays and the associated iQue kits for measuring antibody internalization, ADCC, ADCP, and then also to explore that ADC activity of the Kadcyla. We then also looked at Fc gamma receptor interactions on the Octet. So obviously this gives a good indication of potential Fc function. So starting with the iQue live cell binding assay. So this is a really simple two-step labeling assay where we started with target cells. So in this case, that was highly HER2 positive AU565 cells from a breast cancer cell line. These were incubated with those anti-HER2 antibodies, which were of course unlabeled. And then they were secondary labeled with a fluorophore conjugated antibody. And then we measured the increase in medium fluorescence intensity to give an indication of that binding, expecting that with the anti-HER2 antibodies on the HER2 positive cells to see an increase in MFI with increased antibody concentration. And the key thing about this assay is that all the labeling steps were carried out on ice in order to inhibit any internalization and to ensure that we were really just looking at that surface binding. And this is the kind of data that was generated. So this is a 384-well heat map pulled from the Foresight software. And so here we had 12 different concentrations per antibody with 3 replicates at each concentration. And you can see we had the an IgG control, then we had our trastuzumab, our Kadcyla and pertuzumab. But as well as the sort of native forms of each of those antibodies, we did also include heated forms of the antibodies. So these had been heated at 69 degrees Celsius for either 45 or 90 minutes. And we hope this would also give a bit of an idea of the thermal stability of the antibodies. And in the heat map, the darker grey colours indicate a higher MFI, so increased antibody binding. And the real thing we could pick out straight away from the heat map was that for the trastuzumab, whether it was the native antibody or the heated antibodies, the MFI each concentration was relatively similar. But if we compared the native Kadcyla and pertuzumab to the heated antibodies, you could see there was a graduated loss in MFI at each concentration, indicating that there was a reduced potency binding upon heating of the antibodies. And this is really just the same data as in the previous slide, but pulled out into concentration response curves, and also with those EC50 values pulled out below. And really this just highlights that it confirmed what we saw in the previous slide, with the trastuzumab biosimilar having a fairly constant EC50 value, regardless of whether it had been heated or it was the native antibody. Whereas with both the Kadcyla and pertuzumab, we saw a gradual increase in the EC50 to indicate that reduced potency binding, up to about a 2-fold loss in binding potency by that 90-minute time point compared to the native. And as I mentioned earlier, we did also use this to have a look at the specificity. So in those same wells of the plate where we assessed antibody binding to the AU565 cells, we did also include HER2 negative MDA MB468 cells, and we'd labeled those with the iQue violet encoder dye so that we could distinguish them, as you can see in this plot in the left, from the AU565s. And then here in the bar chart you can see the binding of that top concentration of antibody with those HER2 positive and HER2 negative cells, and it just showed that clearly the antibodies were all highly specific to the antigen positive cell types. So next we had a look at that antigen binding kinetics on the Octet. So as I mentioned earlier, we used the AHC biosensors in order to measure this. So they come pre-mobilized with a antibody that for FC capture of human antibodies, and this meant that we could capture our human FC containing proteins, so the trastuzumab, Kadcyla, or pertuzumab, and that was in the loading step. And then we can measure both the association and dissociation of our analyte, which in this case was the soluble truncated form of the HER2 protein. And this is just the sort of data that was generated. So we loaded a single concentration of the antibody onto the biosensors, so 0.25 micrograms per mil. And then we measured the association of a range of concentrations of that HER2 protein, ranging from 50 down to 3.1 nanomolar. And the trace you can see here is a overlay of 3 replicates per concentration, fit with a 1-to-1 binding model. And so obviously we repeated that for all 3 of the antibodies. And then we pulled out the KD values from the software. And what we saw was that the KD for the trastuzumab biosimilar and the Kadcyla were pretty similar, which was what we expected really, because obviously they both contain the same variable region. So really we're measuring the same interaction between the protein and the antibody. But for the pertuzumab, we did see about a 3-fold increased KD, indicating a 3-fold lower affinity. And if we looked more closely at the association and dissociation rates from which those KD values were derived, we could see that really the association rates were pretty comparable between the 3 antibodies, but actually it was the 3-fold faster rate of dissociation of pertuzumab that was leading to that loss in its affinity relative to the other antibodies. And so then we went back and we wanted to compare how those relative EC50s measured on the iQue compared to the relative KDs measured on the Octet. So what we noticed is that the relative values on each instrument were actually different. So on the iQue, we saw fairly comparable and increased potency binding with trastuzumab and pertuzumab. And then it was actually the Kadcyla that had the lower level binding. Whereas with the Octet, as I've just shown, it was obviously that pertuzumab that had the lowest affinity. And so really we think this just highlights that it can be really useful to generate measurements for target binding using a couple of different platforms. And the instruments provide complementary measurements that really provide lots of information about your molecule of interest. So obviously on one hand, the iQue, you're really getting an indication of the binding of that antibody to the native target as it is on live cells, but you're really also relying on that secondary antibody binding. Whereas on the Octet, obviously the focus is more on that molecular level interaction and the association and dissociation kinetics. But then again, we are also using a solubilized and truncated form of that HER2 protein. So really, yeah, both instruments are going to give some useful data to describe those molecules. So next we had a look at the competition binding using the iQue. So this assay is set up slightly differently to the direct binding assay in that we started by taking a single concentration of the anti-HER2 antibody and labeling with the secondary antibody. Then we mixed those with a range of concentrations of unlabeled anti-HER2 antibody, and then mixed with our target cells. And this meant that we were measuring a decrease in MFI as to give an indication of whether competition had occurred between that labeled antibody and the unlabeled antibody. And so this is the sort of data that was generated for the anti-HER2 antibodies. So you can see here we've got the labeled, a single concentration of the labeled antibodies along the top here. And then we've got a range of concentrations of each of the antibodies along here. So essentially we're looking for a lighter grey colour to indicate that competition did occur in those conditions. And what we saw was as we expected. So there was competition between the trastuzumab and the Kadcyla, and obviously as they bind to the same epitope, that's what we expected to see. But there was no competition between those trastuzumab-based antibodies and the pertuzumab, since it binds to a different epitope, and obviously that highlights why it can be used as a combination therapy, for example, with trastuzumab. So next we had a look at glycosylation on the Octet. So as I mentioned before, we used the GlyM and GlyS kits to measure the mannose and sialic acid content of the antibodies. And this was done using a one-step direct binding assay. So in this, we could dilute all the antibodies to a single concentration, and then we could measure their binding to these lectin premobilized biosensors, which will obviously bind to those specific glycans on the antibodies, and then we can measure the relative level of glycosylation between our antibodies. And this is the data that was generated for the anti-HER2 antibodies. So what we saw was that the antibodies ranked in the same order for both the mannose and sialic acid content, with the pertuzumab showing the greatest level of glycosylation, then the trastuzumab, and then the Kadcyla. And the reason we wanted to measure this is because the glycosylation levels can link to things such as stability. So for example, we did notice that obviously the trastuzumab and the Kadcyla, they are essentially the same antibody apart from those mutations in the Fc region to include the linker to the ADC payload in the Kadcyla. And we did hypothesize that perhaps the mutation to include the linker may result in the loss of some glycosylation sites. And what we did see when we look back at the data from our iQue binding assay was that actually if the Kadcyla had much lower thermal stability compared to the trastuzumab antibody, so perhaps the loss in the glycosylation sites have resulted in lost thermal stability. Although obviously this is going to require some further exploration, because obviously the pertuzumab didn't fit with that theory, showing both high-level glycosylation and sensitivity to the heat denaturation. Also, with the glycosylation can indicate things such as Fc function, but again, I think this is going to require some further exploration and also a larger sample size in order to really start to uncover links between these metrics. So next we use a range of functional assays to assess the antibodies using the iQue. So first we used the iQue Antibody Internalization Kit, which includes a pH-sensitive probe, which you label the antibodies with, and then you can measure the increase in MFI as the internalized antibody with that fluorescent probe starts to fluoresce as the probe enters the acidic, endosomal, and lysosomal pathways. The kit also contains a membrane integrity dye, so you can exclude the dead cells, and then also an encoder dye. So similarly to as was done with the iQue binding assay, you could analyze binding to multiple cell types in a single well. In this case, you could measure internalization with multiple cell types. And what we saw was that the antibodies were fairly comparable in terms of their internalization. But what was notable was that the Kadcyla actually had the lowest potency internalization with the highest EC50, which obviously was interesting because with the Kadcyla being an ADC, it's really the antibody that relies on that internalization capacity to deliver that mechanism of action. And so because of that, we just wanted to explore the ADC activity of the Kadcyla using a really simple monoculture assay with the AE565 cells again, this time incubated with a range of concentrations of each of the antibodies for 72 hours. And then we lifted the AU565 cells from the plate and analysed them using a membrane integrity dye to quantify the level of cell death after that 72-hour period. And what we saw was that, as we expected, only the Kadcyla had induced cell death in that monoculture format over that time period. So confirmed that it did have that unique ADC activity. Next, we wanted to have a look at the ADCP activity of the antibodies, and so use the iQue ADCP kit. So this relies on measuring the co-localization between your target cells and phagocytic immune cells. And so in this case, we set up a co-culture of PBMCs at a 20:1 effective target ratio with the AU565s. The AU565s were labeled with the green encoder dye from the ADCP kit. The dead cells were excluded using a membrane integrity dye, and then the phagocytic immune cells, so monocytes or macrophages, were labeled using the ADCP detection reagent, which labels those CD14 positive effectors. And the kit also comes with a preset gating template, which is imported directly into the software, and that looks like this. So we gate for obviously the entire cell population, and then unique to this kit compared to some of our other kits, we don't include a singlet gate so that we can ensure that we capture those co-localization events. Then we'll gate for the live cell population, and then within that we'll gate for our encoded targets, and then within that we'll gate as well for the CD14 positive phagocytic cells, so that we've got a measure of those co-localized targets and effectors. And this is what the data looked like for our anti-HER2 antibodies. So again, they were pretty comparable in terms of their induction of ADCP activity, but it was noticeable that the trastuzumab did have slightly increased potency compared to the other two, at about 2.5-fold increased ADCP activity. So finally, we had a look at the ADCC activity of the antibodies using the iQue NK killing kit. So for this assay, we co-cultured the AU565 cells again with natural killer cells, and that was at a 5:1 effective target ratio. In this kit, there is a violet encoder dye to label the targets to distinguish from the immune cells. Again, it contains a memory integrity dye, and then it also contains an antibody cocktail. So this allows us to phenotype the NK cells, so the CD3 minus, CD56 positive population. We can then also measure the CD16 expression, so that's obviously a strong indicator of ADCC activity. And then we can also look at the NK activation, so using markers CD69 and CD25. The kit also contains Q beads for cytokine detection. So this is a 2-plex screen containing interferon gamma and granzyme B measuring Q beads. So these are obviously strong indicators of NK activation and also NK killing. And this is the gating strategy provided with the NK killing kit. So it can be imported directly into the software and is provided with the kit. It also comes with a preset compensation matrix. So obviously you don't have to perform any compensation yourself, which drastically speeds up the time during your assay development process. And basically at the top here, we've got our cell gating. So from the cell population through to gating our live effectors, and then distinguishing those NKs and then gating the activation markers on those NKs. And then below here, we've got our Q-bead gating to show how we distinguish the interferon gamma and granzyme B signals. Usually, for example, if we were working with suspension target cells, we would multiplex the analysis of the cells and beads, and so distinguish all of this from a sample taken from a single well. The reason we've had to split this out today to separately analyze the cells and the supernatant cytokines is because of the use of the adherent target cells, so those AU565s. So obviously we need to take a sample of our supernatant out and transfer that to a separate plate before we can then start to remove the media to lift the AU565 cells from the plate for the downstream cell analysis. And so this is the data that was generated for the ADCC activity. So again, the antibodies look pretty comparable in terms of the induction of cell death. But if we do look a bit more closely down at the EC50 values to indicate the potency of the induction of cell death, we can see that actually there was a slightly reduced potency with the trastuzumab biosimilar. And the reason I call this out, despite it being quite a small difference, is that if we then have a look at the CD25 activation marker expression and also the induction of interferon gamma, so a cytokine indicative of activation, we can see that both of those were slightly reduced with the trastuzumab biosimilar relative to the other two antibodies. So, particularly perhaps if we looked at this over a time course, we may have seen that these effects started to have an impact on the amount of killing. I haven't shown the data here, but both the CD69 expression and the granzyme B release were both also reduced with the trastuzumab compared to the other two antibodies. And then here we've also got the CD16 expression, so that was fairly comparable again between the three antibodies, with the decrease in CD16 expression indicating an increase in ADCC activity. And here this slide just really summarizes all of the EC50 values from each of those iQue cell-based functional assays that I've just shown. Really just to highlight that obviously each of the antibodies is going to have a different potency of effect in each of these effector mechanisms, and that it's going to be obviously a combination of all of these mechanisms that's going to induce the overall tumor cell killing activity in vivo. And that's why it's really useful to use in vitro assays such as these in order to pull out the individual activity in order to inform those later in vivo studies. And then finally, we had a look at the Fc gamma receptor to antibody interactions. And for these experiments, we used a streptavidin immobilized biosensor. And this meant that we could immobilize a biotinylated form of CD16A or Fc gamma receptor 3A protein. And then we could measure the kinetics of binding with a range of concentrations of our anti-HER2 antibodies. And due to the nature of the binding with the CD16A protein, in this case, we fit with a 2-to-1 heterogeneous ligand binding model. And this meant that we generated 2 association, 2 dissociation, and 2 KD values per antibody for the binding to CD16A. And so this is the data that was generated. So the key thing we really picked out is that both KD1 and KD2 for the trastuzumab biosimilar were lower, so indicating increased affinity for the trastuzumab to the Fc gamma receptor. When we look more closely at the association and dissociation rates, we can see that with KD1, it was really a slower rate of dissociation that was contributing to that enhanced affinity, whereas with the KD2, it seemed to be more a combination of faster association and slower dissociation that was resulting in the enhanced affinity there. And what we want to do is we want to look further at this, and we want to see how the Fc gamma receptor binding may relate back to the ADCC activity. And to do this, we really want to increase our sample size and also get some sort of more extremes of function and binding. And so to do this, we're going to bring back the heated antibodies that we used in the early iQue experiments. And this just shows some preliminary data from that. So here is a single concentration of the antibodies looking at their Fc gamma receptor binding. And we can see that there's a clear drop-off, a gradual drop-off as well with the increased heat time compared to the native Kadcyla. And we hope that this would obviously relate back to a drop in ADCC activity when we measure back in the iQue assay. So to conclude, the iQue Advanced Flow Cytometer and Octet BLI label-free analysis systems can be used in parallel to broadly characterize antibodies and to generate complementary data sets to describe your antibodies of interest. Both platforms use streamlined and easy to follow workflows, which are coupled with high throughput data acquisition and provides a simple and rapid solution for antibody profiling. The techniques have the potential to screen binding and functional characteristics of libraries of antibodies in minimal time, and so this delivers enhanced drug discovery processes. If you'd like any more information about any of the assays you've seen in this webinar, please visit our website. So we've got web pages and application notes to describe both the iQue and the Octet applications. Thank you very much for listening. I look forward to answering your questions.
The live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in from our audience. Our first question, Kirsty, as some of the functional assays required the use of primary immune cells, is it likely that there would be differences in response between cell donors?
Yeah, so a lot of our work that we've done, particularly with the ADCC and the ADCP assays, has shown that there can be quite a big difference between immune cell donors in terms of the level of response. So you might want to try a couple of different donors. But that being said, typically, although the kind of maximal response might differ, metrics like the EC50 tend to remain quite similar. So it can be fine when you're kind of trying to compare different antibodies.
Thank you so much. And could the Octet Fc gamma receptor assay be used to measure binding to other receptors aside from CD16A?
Yeah, so since we were using the streptavidin immobilized biosensors, we could basically have immobilized any biotinylated protein onto the sensor. Obviously, we'd have to just carry out some further optimization steps. We could have also flipped the assay and carried it out in a more similar way to the antigen binding assay. So instead, immobilizing the antibodies onto the AHC biosensors and then measuring binding of soluble Fc receptors.
Thank you so much. And again, I want to thank our audience for these great questions. Would it be possible, Kirsty, to measure the concentrations of more or different cytokines, and then if in gamma and granzyme B using the ADCC assay?
Yeah, so with the NK cell kit, there are some companion QB kits. So basically, you can just add a few extra beads into the wells, and it means you can directly measure other cytokines alongside the interferon gamma and the granzyme B. Or because the volumes needed for the kit are so low, you would also have plenty of leftover supernatant if you wanted to kind of take 10 microliters away and do a completely separate QB analysis in a different plate.
Thank you so much. And it looks like we have one more question coming in. How could you gather temporal data for the iQue functional assays? Would that require multiple plates to be set up?
So if you're just using sort of like suspension cells, or maybe in the shorter term adherent cell assays where we don't allow the cells to adhere to the plate because we don't leave them for long enough, then you could just take 10 microliter samples over time, and we find this to be non-perturbing to the assay. But in the case that you did want to do kind of a longer term adherent cell assay where the cells have adhered to the plate, you're obviously going to have to lift these off at the end point. So in this case, you would need to conduct analysis of multiple identical plates if you wanted to get some temporal data.
Kirsty, I want to thank you for your time today and your presentation. Would you like to provide any closing remarks for our audience before you go?
Just thanks very much for listening. I hope it's been insightful for you.
Well, again, I want to thank you for your time and your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Sartorius, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed for 2 years until February 22nd, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.